Hi friends, so today we are going to do a food test lab, and this allows us to tell what kind of biomolecules are in different types of food. I'm going to show you how to do the positive and the negative controls for these. So we are going to start with the test for fat, and we're going to actually combine two tests here. So what I need to do is I need to set up a positive and a negative control. In order to make the negative control, I am going to add 1 squirt of a liquid that I know does not have any fat in it. And then to make the positive control, I'm going to add 1 squirt of olive oil. I know that it has fat in it. Now, anytime we are using a pipette, what we're going to do is we're actually going to pipette up and down. And that is going to make it so that we make sure that we have a fairly mixed liquid. So you can see here, this is not pure olive oil. And so as I pipette up and down, it's actually mixing this solution. And so I want to have a fairly mixed solution, which I have now. And I'm going to add 1 squirt of this to my positive control. So what I'm going to do now is I'm going to add some ethanol. This is just pure ethanol. And I'm going to add that to both my positive and my negative controls. I want about 2 to 3 milliliters. I want about 2 to 3 milliliters of this. So I'm going to add a big squirt to both of these. This is for our emulsion test, but we are also combining this with a Sudan 4 test. So we are also going to add about 6 drops of Sudan 4 to both of these. The next step is to cover with your thumb and give it a really good shake. Now that those are shaken up, it's emulsified, and so we can see that our positive and our negative already look different. The final step in this test is to pour this over distilled water. So we're going to do something called decanting. So the first thing that I need to do is I need to get some distilled water. Again, 2 to 3 milliliters of this. I'm going to get some distilled water, and then I'm going to decant. So decanting is when you pour it gently down the side. So I'm going to pour from the positive or negative control into the distilled water. This is the negative control. And so I'm going to pour this gently down the side of the other test tube. And then I'm going to set this one off to the side. We're going to repeat this with my positive control, and I'm actually just going to give it another really good shake just in case, because it seemed like it had separated a little bit. And now I'm going to pour this one. All right, what we should see is in the negative control, it should be clear. So do you see how it's transparent, you can actually see through the liquid? Now it's also stained sort of a very faint light pink color, and that's from the Sudan 4. On the other hand, in our positive control, you can see that there is a layer here that is turned red, and it is sitting on top of the water. That red layer also is not transparent. So you can see through the water layer. You cannot see through this fat layer or the ethanol layer. In our negative control, the ethanol that we put in there, it's mixed, it's distributed through here, and so you can see through all of it. But when there's a fat present, it becomes milky, and then it also stains the fat red. So we're seeing there a layer, and it is stained red. You can also kind of see that there are some bubbles along the side of this test tube. They're just really hard to see. Okay, that is our Sudan and emulsion test for fats. Next, let's test for protein. The test for protein is pretty simple. We're going to use something called biurettes. This is the normal color of biurettes. So I'm going to use this tray where I have positive and negative labeled. And what I'm going to do is I'm going to take about 5 drops of my negative for protein sample. And then I'm going to take 5 drops of my positive for protein sample. And I've put those into the positive and negative wells. The next thing that I'm going to do is I'm going to add about 3 to 4 drops of my biurettes to each of these. This test happens really fast, so we're actually already able to see the results. So I'm going to have to tilt this. What I can see here is that my positive has turned this violet color, and my negative has stayed this pale blue color. So that is the test for protein. If there is protein, then it's going to turn this violet color. And if there is not protein, the negative, it's going to stay this pale blue color. We're going to keep using this tray in order to do our test for starch. So I'm going to now use the other side. It's also labeled with positive and negative. And I'm going to add my negative for starch sample. So I'm going to add about 5 drops. And then my positive for starch sample, again, I need about 5 drops. And for this, we are using iodine. Iodine will stain your clothes and it will stain your skin. So I'm going to be careful not to get this on myself. And I am going to add 2 to 3 drops of iodine to both of these. Okay, for this one, what I am looking for is in my positive, do you see how this has turned like a dark blackish brown color, whereas this is still a more yellow brown color? So the negative doesn't actually change color, but the positive should turn like a black brown color. If there is starch present, it's going to dye that starch. Okay, we're almost done. We're on to our last test. This is the test for sugar. And specifically, these are what are called reducing sugars. They're like monomers of sugar. So we're talking like glucose and fructose. So for this test, we're going to use these test tubes with caps. And as always, I'm going to add about 1 squirt of my negative for sugar, and then 1 squirt of my positive for sugar. The indicator that we are using for this test is called Benedict's solution. So I am going to add about 15 drops of Benedict's to both tubes. And at this point, these look the same. So I'm going to put the caps on these. And then in order to make Benedict's work, it needs to be heated. So I'm going to pop these into a hot water bath for about 2 to 3 minutes, and then we'll come back and check on them. Okay, so we are back with our Benedict's test, which is our test for sugar. In our positive for sugar sample, you can see that it has changed to this orange color. It could, depending on the amount of sugar, it could actually turn a real brick red color, but anything that has turned towards like yellow, orange, red would be a positive sample. This is the negative control. As you can see, it has not changed color at all. That means that there is no sugar in this sample. Okay, those are our food tests. I hope you've enjoyed this. You are going to have to be able to use these in order to test an unknown food to see what biomolecules it contains.